always tell my students and postdocs, this is the best time to be in cancer research. We're rapidly moving these new discoveries into real treatments for patients.
I was stage 4 de novo, or from the start. I had 7 tumors in my right breast, and my cancer had spread to my bones in 5 places.
I was shocked. Leukemia? What is leukemia? The platelet count was so low that she told me I had to go back to Houston. But she wouldn't let me get on a plane because I wouldn't survive the flight.
2 months later, they go in for exploratory, and I was full of tumor. I had over 175 tumors.
We followed the traditional protocol for treating ALL, and she relapsed. And this time, not only in her bone marrow, it was in her spinal fluid.
We were ready to snap if you looked at us wrong. I mean, we were shaking, we were scared.
I think that was worse than the first diagnosis because you'd gone through hell and you're thinking the end is in sight, and now you know what you have to go back and do again for 2 more years.
It was pretty tough. When you hear a diagnosis of cancer, you kind of sit back and you wonder, why me?
I needed that sisterhood that I was missing, and I wanted to join. I thought I could just pick up the phone and get a national organization for African American women, but none existed.
Times are different now, certainly, but at the time that I was diagnosed, people would think that I had received a death sentence.
I spent my days crying. It's the thoughts that go through my head and the worry that I have for my children. And how long do I get with them? How many birthdays do I get to see?
The federal investment in cancer research is critical to cancer care. Almost everyone has been touched by cancer, whether it's a family member, a friend, or themselves.
And so the best part of responding to that is investing in treatment, but we can do so much more.
We've made great strides in the last 5 to 10 years. We're breaking barriers that we never thought possible before. Immunotherapy is one.
We've seen over 30 FDA approvals in 7 years for immunotherapy of different cancers, and we're just starting out.
I was always convinced that I could beat this thing. They said I might be a candidate for this special immunotherapy, what we'll call now CAR T-cell therapy.
They were doing a trial, and they wanted to collect Tori's T cells prior to starting the bone marrow transplant preparation. But if she didn't get into remission, which was a requirement for bone marrow transplant, this would be a backup. It was a study that was just starting.
One of my team members was the lead physician in apalutamide. I joined the phase 2.
AG-221 was the medication I took in my first clinical trial. The doctor decided that it was time to look at other options, and those other options were stem cell transplant. And so that was also a clinical trial. That was the second one. So my son was my donor, and it was called the Belkin study. As I understand, I'm the first one to have that procedure done in the United States.
He says, have you ever had Y90? I said, no. He's like, because you're like the best candidate for Lutathera, the PRRT program that we're going to be doing.
Knowledge is power. We strive to change the mindset of our community. We know that clinical trials will give us the future treatments and change the face of breast cancer for African American women.
I was put on a PARP inhibitor 2 months after I started Lynparza. The breast cancer that spread to my liver is less active and has shrunk in size.
Immediately after I got the T cells, I went to Dave & Buster's, so I was already, my energy level was already like higher than it was before.
Yeah, that's how good she felt.
That's how good I mean, that was the difference between T cells and chemotherapy.
With Lutathera, I'm able to just play with my kids and be a mom.
Very active playing tennis, and now I play golf. I just have not let my chronic disease affect my lifestyle.
I feel good when I go to the doctor. My hemoglobin is up, all my blood counts are up, and I'm a living proof of the fact that a clinical trial saved my life.
Patients are living years, not weeks and months anymore. And so we need ways in which we can have the best possible quality of life long term. So survivorship research is very important to making sure this happens. Without basic science, we would not be where we are today with all of these wonderful developments that have led to real treatments for patients that are giving long-term durable responses.
So we're at a point of time now that an infusion of money and in funding is pretty critical to move this faster.
We have a lot of very smart people taking those funds and figuring out all the genetics and all of those things that researchers do, developing treatment, developing protocols, all working for the same goal to eliminate cancer.
We really don't, as a priority, understand how important the return on investment is. Our priority should be this: we can extend people's lives, their quality of life, and everybody who loves and works with them have a better life by making these investments.
I have to know when my time is up that I have done everything I can to raise money. We need better treatments to keep us alive longer and give us a better quality of life.
Thank you, everyone, for being here, and good afternoon. On behalf of the American Association for Cancer Research, I'd like to welcome you and thank you for joining us in this important briefing. My name is Marge Fody, and I have the honor and privilege of serving as the Chief Executive Officer of this amazing organization, which is really the first and largest professional organization ever to be formed in the world dedicated to advancing cancer research and its mission to cure and prevent all cancers. I thought the video was spectacular in pointing out the importance of research, and I wanted to say that my own sister has benefited from cancer research. She's a 20-year late-stage ovarian cancer survivor, and we are so happy to have her with us in our family. And she was able, because of research, to be able to meet all of her 8 grandchildren. So over the years, the AACR and its more than 40,000 members in the United States and 119 countries around the world have really been at the forefront of every major breakthrough against cancer. Our members are the collective brain trust of all scientific areas in cancer research that ranges from population science, early detection, prevention, cancer biology, translational research, and clinical studies, and all the way now, thanks to Dr. Jaffe's work this year as our distinguished president, survivorship research and convergence cancer science. We marshal the full spectrum of expertise in cancer research to identify the top scientific priorities. We convene conferences and workshops all over the world. We publish 8 high-impact peer-reviewed scientific journals, produce a magazine for cancer survivors and their families and loved ones, and work to raise funds, federal funds for cancer research, as well as from the philanthropic community. The AACR also actively communicates to legislators about policy. Our policymakers are very important to us, and we are so happy that we are also able to educate the broader public about the enormous value of cancer research and the related sciences in saving lives from cancer. And that is why we are here today. A cornerstone of our communications to Congress and the public is our annual Cancer Progress Report, and we're extraordinarily proud today to say that we can share with you the 8th edition of this publication, which we understand all of the legislative offices really look for every year now around this time of the year. As our report details, and as you will hear later in the program, 2018 has been another productive year in cancer research as scientists continue to discover new ways to target cancers that were previously hard to treat and cure. One of the exciting areas of cancer treatment you will hear more about momentarily is immunotherapy. In the past decade, the number of immunotherapeutics has increased almost 5-fold. And the number of cancer types that can be treated by at least one immunotherapeutic has more than tripled. So it's really spectacular to be able to report on that. With the immunotherapeutics that are currently being used to treat patients, including CAR T-cell therapies, which involve using the patient's own immune cells to treat their cancers, we have entered a new frontier in cancer treatment. And I'm glad that I'm the CEO of this organization at a time when I can say that it is just fantastic. And this remarkable advance, such as one of them actually, CAR T-cell therapy, and other novel therapies that we'll talk about today, really would not have been possible without the generous federal funding from the National Cancer Institutes and the National Institutes of Health. This funding is truly the lifeblood of cancer research and biomedical science. The progress that is currently delivering hope to patients also depends on the consistent and sufficient federal funding of the FDA and the Centers for Disease Control and Prevention. In addition, and just as importantly, our continued efforts to accelerate the delivery of new therapies to the clinic is dependent on the participation of cancer patients and survivors in research, and certainly including those who are here today, including their willingness to enroll in cancer clinical trials, to share their personal stories, their health information, and to advocate alongside of the AACR and its members about more funding for cancer research, and also to make effective policy changes. We're particularly grateful to the 12 individuals and their loved ones who have generously shared their experiences about cancer this year in our progress report, because their personal stories put a face on the struggles and the triumphs of individuals with cancer that occur every day throughout this country and indeed the world. 3 of the cancer survivors featured in the report are on our panel this afternoon: Chuck Dandridge, Tori Lee, and Lori Trotman, and we look forward to hearing from them in a few moments. I also would like to recognize other individuals and their loved ones who are featured in the report and who are with us in the audience today. Greg On, Mike Delia, his wife Betty Ann, and their son Mike, Nicole DeCamillo, Karen Jackson, Ron Scolamero and his wife Lori, Johnny Dandridge, and Dana and Chris Lee. At this special briefing today on Capitol Hill, I would like to specifically acknowledge and thank Congressman Mark DeSaulnier and Congressman Ted Poe, both of whom shared their unique perspectives in this report. You see, cancer touches everyone, including legislators. In addition, we thank Congressman Poe for joining us here today, and I understand he'll come in about a half hour. Lastly, I want to thank Congressman Kevin Yoder for securing this beautiful room for us for this event, and for the outspoken advocacy and his strong support for biomedical research and funding for the NIH. Now, beyond the enormous progress in cancer research, 2018 has really been a pivotal year in terms of congressional support for conquering this devastating disease. Following 2 consecutive years of significant increases for the NIH for the first time in more than a decade, Congress took its commitment to the NCI one step further with a remarkable $3 billion increase in the final FY 2018 bill earlier this year. We especially thank congressional leaders Senator Blunt, Senator Murray, Congressman Cole, and Congresswoman DeLauro and their colleagues for their extraordinary bipartisan efforts on behalf of Americans who are touched by cancer, either directly or indirectly. And, as you know, that's all of us. Thanks to their shared vision and dedication, the NIH budget has grown to an astounding $7 billion, or 23% since fiscal year 2015. And now, thanks to the Senate's passage 3 weeks ago of the annual spending bill that funds NIH, another $2 billion increase for the NIH is within reach. We urge Congress to complete its work and make this proposed funding a reality. Also, the National Cancer Moonshot is now entering its 3rd year and is funding promising research in areas identified by Dr. Elizabeth Jaffe, who is here today as our distinguished president, and members of the NCI Blue Ribbon Panel. Thanks to the funding provided by Congress through the 21st Century Cures Act, these projects will speed progress for the benefit of many of those whose lives will be affected by cancer. Although we celebrate the enormous scientific progress and congressional support for cancer research, we cannot forget that this year alone, over 1.7 million Americans will hear the words, "You have cancer," and an estimated 609,000 Americans will sadly succumb to this disease. And this is despite all our work. And of course, had we not been doing this work, the situation would have been worse. These statistics are not just numbers on a page. They are real. They are people. They represent mothers, fathers, daughters, sons, grandparents, friends, and national leaders like Senator John McCain, who just passed away on August 25th after battling one of the most lethal brain cancers, glioblastoma multiforme. We must redouble our efforts not only for cancer patients, but also for all whose lives are touched by this terrible disease. We also must not forget that racial and ethnic minorities, as well as the medically underserved populations, continue to suffer disproportionately from certain cancers and also have higher morbidity and mortality rates. These statistics are unacceptable. It is our responsibility to change this picture, and we can and we must do better. The AACR is committed to altering the trajectory of the devastating collection of diseases we call cancer. And you know there are more than 200 diseases we call cancer and many hundreds of subtypes. So we have a lot of work to do. So working together, we can take major strides toward realizing the goal of fundamentally changing the face of cancer, of this devastating disease, at the earliest possible time. Now I'd like to turn the podium over to Dr. Jaffe, but before giving her the microphone, I'd like to introduce her appropriately. It is my great honor to introduce her as our esteemed president of the AACR. She is Deputy Director of the Sidney Kimmel Comprehensive Cancer Center at Johns Hopkins in Baltimore. She is a world-renowned expert in the hottest area of cancer research today, cancer immunotherapy. She has conducted pioneering research that has had and continues to have an enormous impact on the discovery and early clinical development of investigational immunotherapies for several different cancers, most notably breast and pancreatic cancers. In addition to her extraordinary science, Dr. Jaffe is also a distinguished, highly effective leader in cancer policy at the national level. She is the U.S. presidentially appointed chair of the National Cancer Advisory Board. I think that's you're the first woman who ever had that position. Okay, all right, you go, girl. And, and she served as co-chair of the National Cancer Institute's Blue Ribbon Panel for the National Cancer Moonshot Initiative, which was convened to provide expert advice on the scientific goals and implementation of what has now been named the Beau Biden Cancer Initiative. Her expertise and vision are invaluable to the AACR as we work together this year toward our shared goal of defeating all cancers at the earliest possible time. Please welcome Dr. Jaffe to the podium.
Thank you, Marge. That was quite an introduction. I hope I can live up to it today. Good afternoon, everyone, and I want to thank you all for joining us here today. I am delighted to be here to discuss the AACR Cancer Progress Report 2018, which details how research is powering progress against many forms of cancer. As you all know, it really is an incredible time to be in cancer research. And although I can talk about the rapid pace and progress, I think you all heard from the most important group of people, the survivors, on the video just now, and you will hear from survivors on this panel what we are able to do with research these days. It is turning into real treatments for our patients. Importantly, as our report underscores, the scientific discoveries that have enabled us to develop better ways to prevent, to detect, to diagnose, and to treat many cancers are largely fueled by the federal investments in research through the NIH and the NCI. And we want to thank all of the congressmen and women who have supported this and their staffs over the years and for the increased funding that we're continuing to get so that we can continue this great progress. During the almost 30 years since I began my fellowship in oncology, I have witnessed unprecedented progress against cancer. Many advances are a direct result of progress in basic research and the persistent pursuit to better understand cancer biology. In fact, when you look at immunotherapy, it took 30 years to understand how the immune system recognizes and kills cancer before we had these great drugs that are now turning deadly cancers into chronic diseases and, in some cases, cures. So continued investment in both basic and clinical research will allow us to make major headway, moving forward so that we could affect more patients and give them better lives. Just consider the progress we've made in the past quarter century. Between 1991 and 2015, cancer death rates fell by 26%. This equals 2.4 million lives saved. An estimated 15.5 million Americans, more than ever, are living through and beyond a cancer diagnosis, and they are living fuller and higher quality of lives, and that's our goal. While not the only measure of success, the report documents the significant progress we are making year after year against cancer by highlighting the medical products that have been approved by the FDA over a 12-month period. Consider this snapshot of just 1 year, just this past year. Between August 2017 and July of this year, the FDA approved 14 new anti-cancer therapeutics, new uses for 11 previously approved anti-cancer therapeutics, and 1 new surgery guiding system. Development of these new tools for cancer treatment is possible because of the tireless work of individuals across the spectrum of cancer research, from basic to translational to clinical care, to public health, and to, again, cancer care. It is also possible because of cancer patients themselves who are willing to participate in clinical trials. And again, you heard from the video the importance of that, and you'll hear today again from people on this panel how their lives were saved because of clinical research. These trials play an indispensable role in advancing progress against cancer, and patients participating are part of our team in making this happen. I will focus my remarks today on 3 areas. Innovation, specifically, I'd like to discuss new targeted therapies and advances in immuno-oncology in the past 12 months, just as a few examples of what we're seeing today. I also want to talk about cancer health disparities. Specifically, I want to give some examples of how past advances have not benefited everyone equally, and certain segments of the US population shoulder an uneven burden of the diseases called cancer. I also want to talk about survivorship. Survivorship more and more is becoming an important aspect of not just how we treat patients post-therapy, but we need research on survivorship to know the best way to treat them from the start of therapy. We all know a cancer diagnosis is life-changing, and we have a responsibility to not only help people survive cancer, but to help them thrive after cancer treatment ends. So let's start with innovation. So decades of basic research, including ambitious, large-scale, federally funded initiatives like the Human Genome Project, have fueled a revolution in our understanding of and approach to cancer treatment. We discovered that genetic errors or mutations are the drivers of cancer initiation and progression. This discovery set the stage for the new era of precision medicine, an era in which the standard of care for many patients has changed from a one-size-fits-all approach, which was not working very well, to one in which greater understanding of the patient and his or her tumor dictates the best treatment option for that patient. Through advances in genomic research and by utilizing cutting-edge DNA sequencing technologies, we are developing diagnostic tools to detect genetic errors and better predict which treatments will help which patients. We're also better able to develop new targeted treatments that are both more effective and less toxic than the treatments that were the mainstay of cancer care even just a few decades ago. 9 of the 14 new therapeutics and 6 of the 11 previously approved therapeutics highlighted in the report fall into this category of what we're calling molecularly targeted therapies. These advances mean that we now can apply precision medicine in the treatment of an even wider array of cancers, including some that arise in blood-forming tissue, such as the bone marrow, or in the cells of the immune system. These blood cancers are broadly called hematologic cancers. One hematologic cancer, acute myelogenous leukemia, or also called AML, is projected to be the second most common type of leukemia diagnosed in the United States this year, and is the leading cause of leukemia-related deaths. Until last year, treatment of AML had changed little since the 1970s. Now, because of research, we have substantially increased our understanding of both the biology of AML and, importantly, the genetic mutations that promote leukemic generation. As a result, there are now several new therapeutics that target types of AML that are driven by specific mutations. These new therapies are helping patients like Chuck, who you will hear from soon, achieving long-term responses and being able to resume a relatively normal life. In the coming years, as cancer genomics research continues, we will further refine the way we use precision medicine, and we will expand the use of molecularly targeted therapy to more forms of cancer. I'd now like to just spend a minute or two talking about immuno-oncology. One of the most exciting new approaches to cancer treatment is immunotherapy. The field of immuno-oncology has, after decades of work, and frankly, I've been in it for 30 years, so it's very exciting for me to see all this progress, but it took 20 years of work by many different people around the world to get to this point. It's now opened up an entirely new strategy for fighting cancer. Immunotherapies have unleashed the power of the patient's own immune system to eliminate cancer, much in the way that the body is able to fight viruses or bacteria. One of the most exciting developments is that many cancer types can be treated effectively by the same agent. Patients with late-stage cancers have experienced long-lasting responses. This provides more patients with longer and healthier lives. We've especially seen this with the class of immunotherapeutics called checkpoint inhibitors, which release the brakes on specific immune cells so that they can attack and eliminate a cancer cell. When the first AACR Cancer Progress Report was published in 2011, only one checkpoint inhibitor had been approved, and only for one type of cancer, malignant melanoma. It took 3 more years, but when our 4th report was published in 2014, a second checkpoint inhibitor has been approved. Today there are a total of 6 of these agents that are on the market for different cancers. More remarkable than the tripling of the number of FDA-approved checkpoint inhibitors is that now 1 or more of these immunotherapeutics has been approved for treating 12 different types of cancer and for treating any type of solid tumor characterized by the presence of a specific molecular characterization. As Dr. Foti noted, last year the FDA approved CAR T cells for children and young adults with acute lymphocytic leukemia, also known as ALL. And like Tori Lee, who's here with us today, and she's here with her parents, she has now been hopefully cured of this disease. ALL is the most common cancer diagnosed among children ages 0 to 14 in the United States, with more than 3,000 new cases projected to be diagnosed again this year. The development of CAR T-cell therapy is groundbreaking and life-transforming for patients like Tori and patients with non-Hodgkin's lymphoma like Mike D'Elia, who is also here today in the audience. CAR T-cell therapy is a complex medical procedure in which the immunotherapy is customized for each patient. During treatment, immune cells are taken from a patient's blood. They're expanded in a culture dish to the numbers needed. They're genetically modified in the laboratory, and then they're returned to the patient where they can now attack and hopefully kill that patient's cancer. The basic, translational, and clinical research that led to the CAR T-cell therapies are now saving lives, and this could not have happened without federal funding. Next, I want to talk a little bit about cancer health disparities. This is a very important problem that we need to address, and we need to address it now. We need to ensure that everyone benefits from the groundbreaking advances that we are making against cancer. No one is immune from cancer. It can strike anyone of any age, gender, race, or ethnicity, socioeconomic status, geographic location, and even political affiliation. And yet, sadly, we know that the advances highlighted in this report have not been accessible to or benefited everyone equally. The stark reality is that certain segments of the population continue to shoulder a disproportionate burden of cancer, and we have to do something about it because we now have treatments that they can benefit from. For example, African American men have a prostate cancer death rate that is more than double that for men in other racial or ethnic groups. African American women are almost twice as likely to be diagnosed with the worst form of breast cancer, called triple-negative breast cancer. Men living in Appalachia have a lung cancer incidence rate that is 26% higher than that for men living in the remainder of the United States. Women in rural areas are twice as likely to die from cervical cancer than women living in urban areas. And kidney cancer is more common and causes more deaths in Native Americans than in any other group in the United States. These differences in incidence and mortality reflect disparities found across the cancer care continuum for underserved populations, including in prevention and exposure to carcinogens, access to screening and early detection, and treatment and survivorship care. These disparities are unacceptable, and it is imperative that all stakeholders in the research community work together to more fully understand the reasons for them and then identify ways to eliminate them. Finally, I want to just talk a little bit about survivorship. The progress I highlighted earlier is great news because it is allowing more people to survive longer after a cancer diagnosis. Because people are living longer after cancer or with cancer in a more chronic form, we need to invest in more research to identify ways to improve the quality of life for patients and survivors by minimizing the risks of short and long-term side effects from these treatments. We must ensure that the exciting new treatments we are developing confer maximum benefit and minimal risk. We also need to consider how we are using current therapeutics so that they can be employed in ways that give patients the best possible quality of life for the long term, not just for the short term. I want to commend the two members of Congress, Representatives Mark DeSagne and Ted Poe, for forming the House Cancer Survivors Caucus to raise awareness with their congressional colleagues about these very important issues. Some ways that patients and survivors can improve their health and reduce the risk and severity of treatment side effects are by eliminating tobacco use, maintaining a healthy weight, and participating in physical activity. One important area of survivorship research focuses on those diagnosed with cancer as children or young adults. These individuals have the additional challenge of adapting to decades-long cancer survivorship while beginning careers and thinking about starting families. As we will hear from Lori Trotman shortly, being proactive and talking to healthcare providers about the possible long-term and late side effects of treatments before they begin is very important. Many treatments for cancer, including surgery, radiotherapy, and cytotoxic chemotherapy, can cause infertility. Thus, parents of children and adolescents diagnosed with cancer and adults, particularly young adults diagnosed with cancer, should consider discussing with their healthcare providers whether fertility preservation is an option. It is imperative that we accelerate research on survivorship and work harder to improve not just the quantity of years, but the quality of life for the increasing number of people who will become cancer survivors in the United States and around the world. So just to conclude, I want to say that as our report shows, research has and will continue to spur progress against cancer. We need to seize the opportunities that we now have to drive research forward. We can stimulate additional breakthroughs that will save more lives. As we look to the future, I am confident that one way we will be able to accelerate the pace of progress is by increasing collaboration between cancer researchers and experts from other disciplines, such as mathematics, physics, chemistry, engineering, and computer science. The new wave of innovations driven by what we call now convergent science will allow us to integrate and harness patient data to achieve the full potential of precision medicine. In addition, the incorporation of new cross-disciplinary technologies, such as detecting cancer in blood and other fluid samples, and artificial intelligence will provide tools to address a wide range of questions across the spectrum of precision cancer care. To ensure we can do this, we need to continue to prioritize biomedical research as a nation. A major goal of the Cancer Progress Report is to serve as a call to action to Congress, the administration, and even the public to continue to make funding for biomedical research through the NIH a national priority. We urge members of Congress to finish the work that they have begun and enact another $2 billion increase for NIH in the FY 2019 budget. Support for the FDA, a crucial partner on the path from lab to clinic, is also required if we are to get new, safe, and effective therapies to patients more quickly. Likewise, cancer programs at the CDC are essential in the application and implementation of research through programs that benefit people, families, and communities. With the right resources, we can continue our quest to conquer cancer and accelerate the pace at which we develop new strategies to prevent, detect, diagnose, and treat this set of diseases. Most importantly, we can bring hope to patients and their loved ones everywhere. And we are bringing hope. You heard it on the video. We need to continue to do that. And by being able to have the right funding to develop new therapies, we will be able to continue to do that. So with that, I'm going to stop there. And I believe we now have Congressman Ted Poe available to speak with us. So I'm very pleased to introduce the Congressman who represents the 2nd Congressional District of Texas. Congressman Congressman Poe serves on the House Judiciary Committee and on the Foreign Affairs Committee as chairman of the Subcommittee on Terrorism, Nonproliferation, and Trade. Prior to coming to Congress, Judge Poe served for 8 years in the Harris County District Attorney's Office and then for 22 years as a criminal court judge in Houston. Congressman Poe is founding member and co-chair of the Congressional Cancer Survivors Caucus. He graciously shared his personal experience with cancer in this year's Cancer Progress Report. He has been a strong and vocal supporter for medical research as a bipartisan priority for Congress. Congressman Poe, we thank you for being a champion for cancer research and for coming here today and talking to us. Thank you.
Well, thank you all for letting me come in and chat just a few minutes. I know you'll have a lot on your plate today, but it was worth it to me to get on a 6 a.m. flight from Houston this morning and fly here to talk to you, and I appreciate the chance to be here. Yes, I am Ted Poe. I'm from Texas. That's another foreign country. And Mark DeSaulnier is from California, and he thinks California is a foreign country. He's a Democrat. I'm a Republican. We never discussed those issues because when we formed the Cancer Survivor Caucus, it's a nonpartisan issue. As you know, cancer doesn't play favorites. It goes after anyone of any race and of either party. So we formed that caucus to raise awareness about cancer to other members of Congress. That is the purpose of the Cancer Survivors Caucus. We've introduced one piece of legislation so far. It's called the Cancer Care Planning and Communications Act. What does it do? It amends Medicare to require that the main oncologist treating cancer of that patient works with all the other medical professionals when dealing with this cancer patient. All the other medical needs, all the other doctors, so that there is a united effort in treating not only cancer, but treating the patient. And so we hope that passes so that there can be some organization and working together. When Congress passes legislation, and Mark and I both talked about this, we don't want to make things worse. We want to try to make them better. All of you know that sometimes Congress, when we pass legislation, whatever the situation is, it's not improved, it's worse. So we don't want to do that, but that's what this legislation does. Organizes and streamlines the bureaucracy so that the medical profession can do what they do, and that's cure folks. 2 years ago, I was in a parade in Houston, 4th of July parade. Been in it ever since I was a judge. And I felt and looked awful, but according to my staff. And after the parade was over with, I'm headed to Washington to come up here, you know, and save the country. My staffer told me I should take you to the hospital and not to airport. Of course, I said, take me to the airport. Got here, and I just got worse. So saw some medical personnel, got referred to the Georgetown Hospital, came in contact with Dr. Ujani, and she ran some tests, found out that I had leukemia. Leukemia, I couldn't even spell leukemia at the time. I really couldn't. And so she made the recommendation that I go back to Texas. I'm from Houston, and go to MD Anderson Hospital, where they have, I think, probably the greatest, one of the greatest cancer treatment facilities in the world. And so she ran another test, several tests. I did a bone marrow biopsy and said, you can't go
back to Texas yet because you're not going to make it if I put you on an airplane. So you need to go to the emergency room, and you need more platelets. Platelets. What's a platelet? I thought that was a small dinner plate, you know. But she said, you need platelets, or you're not going to make it. So as you all know, platelets have a short lifespan. And so hospital folks called around, and in the middle of the morning, 1 or 2 o'clock in the morning, finally 3 volunteers who I will never know came from around here, went to the hospital, donated platelets, and I got those platelets, and I thank the good Lord for those 3 strangers. Got back to Texas, and I was admitted at MD Anderson for leukemia. It was a rough go of it for several weeks, but thanks to the good Lord and Dr. Ujani and Dr. Katarjan, I'm doing a whole lot better now. But the point being, when something like that impacts one of us personally, it kind of gets your attention in a lot of ways. And one way that it got my attention, since I am a legislator, is, well, what are we doing in Washington about medical research on cancer? And so I did some research on that issue, and it felt like what Congress needs to do is to emphasize the importance of research and do everything we can to finance and fund research in the area of cancer, because the research is having great results, as you know. I'm taking an oral chemo pill. I'll take it for the rest of my life, but just a few years ago, that was not available for leukemia patients. And the life expectancy for the type of leukemia I have wasn't very long. But now, because of research, it's not only helping me, but it's helping other people. So that is what Congress needs to do. And that's what Congressman DeSaulnier and myself are going to push, is that Congress needs to fund more research funds so y'all can do your job. And so that's our goal. And I would just encourage you, if you have suggestions, which you have, you come see us. And we want to expand the Cancer Survivors Caucus to get that message out to other members of Congress so that we can have a better quality of life, and so you can do your job without having to worry about where the funding is coming from to do that very important job. Thank you very much. I appreciate it.
Okay, thank you again, Congressman. That was riveting. It's now my honor to introduce one of my colleagues, Dr. Quinn Lee. She is chair of the Department of Radiation Oncology at Stanford University in Palo Alto, California, where she's also co-director of the Radiation Biology Program at the Stanford Cancer Institute. She also holds the Catherine Dexter McCormick and Stanley Memorial Professorship at Stanford. Dr. Lee's research spans from bench to bedside, focusing on head and neck cancers and specifically on tumors of the salivary glands. Dr. Lee's lab was one of the first to identify circulating biomarkers from tumors and blood that might indicate how patients will respond to radiation. These discoveries have led to the application of some of these markers in clinical trials, testing new treatment strategies. On the clinical side, she has led multidisciplinary, multi-center phase 2 and 3 clinical trials, testing the addition of novel drugs as either radiosensitizers or radioprotectors with chemoradiotherapy in head and neck tumors. Dr. Lee is a member of the Cancer Progress Report 2018 Steering Committee and serves on the Steering Committee for the AACR's Radiation Science and Medicine Working Group. She is the recipient of numerous awards and honors for her work, including the induction into the National Academy of Medicine. Thank you, Dr. Lee, for lending us your expertise and talking with us today. Thank you.
Thank you, Dr. Jaffe. It's my privilege to be here to share some of the progress in radiation therapy and to meet all the survivors who have really share your wonderful stories with us. First, I'd like to thank all the funding agencies for supporting cancer research and all the cancer patients who have been willing to participate in clinical trials. Without your support, we'll not be where we are today. You've heard quite a bit from Dr. Jaffe and Dr. Fodes about the progresses that we've made over this last year. Therefore, I will focus my remarks on 3 areas, specifically advances in radiation therapy, advances in targeted radiotherapeutics, and advances in cancer detection. First, I will talk about radiation therapy. You may be aware that radiation is one of the cancer treatments, but you may not be aware that it's used in more than 50% of cancer patients. It can be used for cure or for decreasing symptom, and as the second most commonly used type of cancer treatment for curative treatment globally, it is cost-effective. In the past decade, the advances in our field has mainly focused on increasing the precision of radiation delivery so we can target the tumor more precisely with less impact on normal tissue. This so-called spatial precision is accomplished through multiple approaches. One of such approaches is the use of computer-aided radiation treatment planning, such as IMRT, that can allow us to pin the dose more precisely to the tumor. The second approach is our ability to visualize the cancer during treatment with imaging devices such as CT or MRI scanner that are directly coupled to the radiation therapy machine. This is known as image-guided radiotherapy. The third is to expand the radiation beams against cancer. The traditional radiation beam is high-energy X-ray, which can deposit much of the energies in the paths toward or away from the cancer. This can lead more dose to nearby healthy tissue, thereby limiting the size and the number of tumors that we can treat safely. Heavy charged particle beams, such as proton or carbon ions, have different physical properties. The electrical charges of these particles, called the Bragg peak effect, where most of the dose is delivered at the end of the particle range, is placed inside the tumor. This physical property allows more precise tumor targeting with less damage to normal tissue. In addition, heavier particles such as carbon ion can create more damage to cancer cell, leading to more cancer killing and more synergism with immunotherapy. Therefore, these type of treatment are potentially more effective against cancer that are resistant to radiotherapy, and we potentially treat tumors that are normally cannot be treated with X-ray. What have these accomplishments accomplished so far? IMRT has resulted in less late toxicity in several cancer types and better survival in certain tumors such as head and neck cancer. MR-guided radiation treatment has resulted in better tumor control for cervical cancer. Since we can target the cancer better and see them during treatment, we're now able to reduce the duration of radiation treatment for many types of cancer. For an example, we can now safely treat some breast cancer over 1 to 2 weeks instead of the 6 weeks radiation course, so the patient can get back to their normal life much sooner. In addition, we can give very large doses of radiation, usually 1 to 5, in an ablative treatment, just like surgery, so we can treat smaller tumor. For this type of ablative radiation treatment can be used to treat early-stage lung cancer that are not amenable to surgery, to treat patients with multiple brain metastases without the whole brain treatment, or can treat patients with up to 5 sites of metastases, also known as oligometastatic disease. Adding this type of ablative radiation treatment to chemotherapy has actually been shown to improve survival in patients with oligometastatic lung cancer. These ablative treatment can serve also as in situ vaccines, so it can synergize very well with immunotherapy that you've heard from Dr. Jaffe, the immune checkpoint therapy. It has been shown to have good results in combined with immune checkpoint therapy in brain metastases, and more recently, oligometastatic lung cancer in randomized setting. Finally, the use of proton beam has resulted in better neurocognitive function and less secondary malignancy in pediatric cancer patients. In addition to technical innovations, Recent advances in radiation therapy have focused on the use of information from tumor cells, such as genomics and proteomics, to identify patients who most benefit from radiation treatment. For example, the use of GenomeDx genomic signature has been now used in prostate cancer to identify patients who benefit from radiation therapy after surgery. The use of circulating tumor cells after surgery in early-stage breast cancer can be used to determine who may benefit from additional radiation to the tumor bed, and the use of tumor HPV DNA in the tumor status can be used to identify patients with head and neck cancer who may benefit from lower radiation doses or less chemotherapy. Next, I'd like to focus on targeted radiotherapeutics. As we learn more about cancer, we can be smarter in developing radio-labeled targeting treatment against them. These are protein fragments that are tagged with radionuclides. These fragments serve as carrier to bring the radiation directly to the tumor cell. 2 new targeted radiotherapeutics have been approved this year by the FDA. I'd like to highlight one of them, which is Luthera, and has been approved for the midgut neuroendocrine tumors. These are the cancers that arise from cells of hormonal and nervous system. They can arise anywhere in the body, most commonly in the midguts. That's been shown to express high level of a type of hormone called somatostatin and are dependent on this pathway for growth. Therefore, a novel approach is to link somatostatin with a potent radionuclides that can release a radiation at very short range, within 1 to 2 millimeters. Luthera is a molecule that linked lutein to somatostatin. In a randomized study, where half the patients were assigned to standard treatment versus standard treatment plus Luthera, the addition of Luthera has led to 6 times more higher response rate, 6 times more patients living without cancer progression for 2 years, and significantly less death. Nicole DiCamillo in the audience has combated this rare form of cancer for years and received Luthera for compassionate use program in 2016. She has continued to experience benefit from this treatment, and using Nicole's old word, Luthera has changed my life. At this moment, the treatment of these drugs is one size fits all, with the same radiation activity given to all patients regardless of size of the tumor or the number of the tumors of the patient. We need a better method to estimate the radiation dose that's actually received by this treatment to the tumor, and more funding is needed for these type of study. Finally, the best way to minimize treatment-related side effects is to detect the cancer much earlier than we can now, when they're small and have not spread. In addition, we need to know when the cancer has become resistant to our current treatment in order to change course. Biomarkers that can do this hold promises of enhancing cancer cure. Liquid biopsy is one of such promises. During the course of cancer development and treatment, tumor routinely shed cells and free DNA into the patient's blood. Recent studies have shown that it's possible to use blood, urine, or saliva sample, the so-called liquid biopsy, rather than traditional tissue biopsy with a needle or knife, to obtain material that can be analyzed to provide information about the changes in the patient's cancer during treatment or even before treatment. Liquid biopsies have the potential to transform detection, diagnosis, and treatment, and is being tested in several clinical trials. In addition to trying to detect DNA or proteins that are naturally made by cancer cells, another approach is to force cancer cells to exist to make a reporter molecule in the blood and urine. In this approach, one would eventually swallow a pill, and if a person has cancer, he or she would urinate our biomarker. This approach is being tested in humans at high risk for cancer starting this year. All these advances have been made from funding support by Congress through the NIH, the NCI, the Department of Defense, Department of Energy, the NSF, and funds raised by cancer organizations such as AACR, ASTRO, and ASCO. As Dr. Jaffe and Fody have urged, we need members of Congress to enact another $2 billion increase in NIH funding. We also need Congress to support the FDA, the CDC, and other funding agencies. We owe it to our patients and their family to accelerate the pace of research to prevent, detect, and treat cancer. I'd like to thank you for your attention. I'd like to ask Dr. Jaffe back to the podium to introduce the next speaker.
Well, the final part of this session, I think, is the most important. We're going to hear from our 3 survivors who have agreed to talk with us today. I want to introduce all 3 of them first, actually more than 3, because one of our survivors came with her parents. I want to introduce all of them to you first, and then we'll hear their stories. The first is Chuck Dandridge, and he lives with his wife of 43 years, Johnny, in Mansfield, Texas, where he is retired after a 36-plus year career I heard about, where he worked in the YMCA. They have 2 adult children and 1 grandson. Following a diagnosis of acute myelogenous leukemia, Chuck participated in 2 clinical trials, one testing a targeted therapy and one testing genetically engineered stem cell transplants. His son was a stem cell donor, and Chuck was the first person in the United States to undergo this procedure, as we heard a little bit on the video. The transplant was a success, and as a result, Chuck has been able to resume an active lifestyle, and we'll hear from him in a few minutes. I also want to introduce Tori and her mom, Dana, and her father, Chris, who's in the audience with us. They're from Ocean Township, New Jersey. Dana is an elementary school teacher and high school track coach, and Chris works in the health and fitness industry. Tori, the youngest of their 4 daughters, was diagnosed with leukemia, and I think you heard, again, a little bit about her. And this was resistant to conventional therapy, and she participated in the original pediatric CAR T-cell trial in 2013, and today she's a thriving 15-year-old. So we'll hear from them as well. And finally, I wanted to introduce Lori Trotman. Lori is a native New Yorker, but 3 years ago she embraced a move to the South to be closer to her family and raised her son as a single mom. A survivor of breast cancer, Lori strives to focus on greater quality of life that is not solely driven by career choices, but being present with her son. Now, I'd like to first ask Chuck to come up, and then we'll hear from Tori and her parents, and then from Lori. Thank you.
Good afternoon. Thank you, Dr. Jaffe. I had a bone marrow aspiration in 2014 to determine where my blasts were at. My blasts were at 8 or 9%. And those of you in the medical profession will understand all this. I still am trying to learn it. But my blasts were in the range of 8 to 9%. And the doctor told me that as I developed the MDS, there was a good chance, or 25% chance, that it could develop into leukemia. So given that, I was told that the only cure for my disease would be a bone marrow transplant. That's what I was told at that time. And so the doctor started treating my MDS with a biological medicine called Vidasa. And I did that for about 5 or 6 months. And while doing that, they also introduced me to a product called Neupogen. Neupogen was to help to increase my absolute neutrophil counts, or A and C is what they called it. During those months, and I went on for about, I think that was about 5 months or so that I had this treatment, maybe a little longer. My ANC did increase, but my bone marrow was so disruptive that it wasn't allowing it to stay up, and so it would drop back down. So in September of 2014, I was suggested I do another bone marrow aspiration, which I did, and in that, my bone blast had increased, my blast had increased to 20%. 20% then meant that I had fallen into the stage of what's called acute myeloid leukemia. So me and my family and I, when I say I, my journey was our journey. It was that of my family, my medical professionals, my church, and all who, helped me through that journey. So always think of it as ours when you hear me say I. But we went to UT Southwestern. That was our choice of hospitals. We had several choices, but we lived close to that area, so we thought that would be the best and not have to put a burden on the family having to move around the country. So we went there. Doctors told me that my only option of cure at that time, they introduced stem cell transplant to me. That's the first time I'd heard that. I'd been hearing about bone marrow, but stem cell was introduced to me. They also talked to me about clinical trials. Clinical trials were something that, you know, when you hear clinical trials, it sounds strange to hear it because, you know, well, maybe this is a first or something that's going on. They don't know the answers to it yet, and that's what it is. But they talked to me about it. They did tell me that I had to have another bone marrow aspiration. I had a lot of these aspirations. I must have had about 12 of them, maybe more. So they became commonplace to me. But I had another bone marrow aspiration, and to determine if I had a specific gene. That gene was an IDH1 or IDH2. And I did have the IDH2 gene, which qualified me to be in one of 2 clinical trials that I would eventually get into, and that was, the first one was to take a medication called AG221, and I can only use it by that name. There probably was a medical name to it, but I remember it just by my pill box saying AG221. I took that medication every morning, like clockwork, as directed, and then the only time I deferred from that time was when I went into the clinic for them to check me. So I did that for about 6 or 8 months. And when I was told about this clinical trial and it was identified that I was eligible for it, I looked at it like this, and I was saying that we didn't know if it would help me, but we knew that the research and the results from that would help others if it didn't help me. Excuse me a minute. Life is good. My family and I, we decided that that I would go on through with the clinical trial, which I did, and taking the medication got my blast down to about 3%. So that was good, 3%, but it didn't eliminate it. And then when my doctor told me I needed to, they would do aspiration periodically, and those blasts would hang around 3 to 5%. So she said, well, it's probably time for us to consider transplant. But the good news was, and I'm one who had never gotten sick. I mean, I never experienced any illnesses or stuff like that. I just went to my normal checkups. The doctor would check me every 6 months, and there was nothing there. But I had this silent disease eating at me, and I didn't even know it. I never, like I said, never got sick. But I did get a virus, a viral infection. I went into the hospital for about 11 days to be treated for that. It was not a cause of the medication. It was really just my immune system being so low that I was just susceptible to those things. I got it. I remember I was out of town, and I drove about 3 hours when I noticed I had gotten sick. And I took a couple of Tylenols, and I called my wife on the phone. We talked on the phone, and she said, just come on home, and I did. And when I got in, she said, the doctor wants you to come right to the hospital. They put me in the hospital for 11 days. I did that, and once I was cured from whatever that virus was, I had been on the AGG2 for several months, and the doctor said I'd been through 2 phases of that product. And they said, well, we've got your blast down to 3-5%, and they're not moving any lower. So it's really time to go forward with the transplant. So having no full siblings, and I was going to participate in what's called the Bellicum study. Having no full siblings, I have 6 brothers and sisters, but none of us are full siblings. So that wasn't a good idea to use them as a donor. But I have, I think my wife and my doctor had talked about this quite often. They talked about the ram in the bush. And that ram in the bush, as you saw in the film, was my son. Doctor had explained to me, all of your children have at least 50% of your DNA. Sometimes more, but at least 50%. So my son was tested, and he had 50% of my DNA, but he also had all the markers needed to be a qualifying donor to me, and that was certainly a plus and very, very good option. So I went into the transplant program for the Bellicum study, and I was given about 4 or 5 days of chemotherapy, and then 1 day of radiation. And the way the doctor explained to me the radiation was that it was like an infant would get radiation. It wasn't a heavy dose, but the chemotherapy didn't do all the things to me that they thought it would. I did lose my hair, but it was coming out anyway. And I didn't have the illnesses and things like that, so I was blessed in that respect, that I didn't go through all of the illness that they had anticipated that I might go through. So anyway, my transplant started in July of 23rd, 2015. And the way they did that, they had taken cells from my son, I think they were called T cells, and they sent those to be engineered down in Houston, and then they came back to the clinic and they were held in storage until I was ready for my transplant. Well, I went in and the transplant started on the 23rd. I had my first round of infusions from my son. I think they took them from him on the 22nd, put them in me on the 23rd. Then on the 24th of July, 2015, they took a second, they put another set of his cells into me. They put the T cells in on August the 6th, 2015. And the way I understand the T cells were that they were put in as a mechanism so that should I get a disease called graft versus host, or GVHD, they would have a mechanism in there to turn that on so that if I got sick with that disease, severely sick with it, they would have a way of combating it. And it would turn it on and it would kill the bad stuff, but in return, leaving the good stuff that was put into my body alone. So as you have to understand, I was completely void of any immune system at one point. So they did that, and I was sitting in my hospital bed, or just sitting up, because they tell you all the time when you have these kind of treatments, they don't want you just laying around. And I can remember my wife making me go and walk and do stuff, and when you don't want to do it, she just made me get up and walk. But I did. And I was sitting in my room one day, and I had what I'm going to call my epiphany. And that's the only thing I could think of. And my wife wasn't in the room, but I called out to her. There were no medical professionals in the room. And I tell the story about being touched on my shoulder twice. And this had never happened to me before. So I said that I believe God had touched me and told me it wasn't my time. I'm a living person that's here because of the therapies that are available now. I have an uncle through my wife's marriage who was diagnosed about 40 years ago with leukemia. And he's still living today. He would call me and encourage me to just hang in there. He didn't go through the modern technology that we're doing now, but he had what was being done at that time. But he is a 40-year survivor of leukemia. God bless him. And he gave me a lot of encouragement, talked to me quite a bit. Matter of fact, I would get calls from him probably more often than I used to, just encouraging me and telling me what to do in terms of how to eat and things like that. And I was appreciative of that. But I knew that going through these clinical trials was important. And the importance of it is that I had received some stories were written on me about it being the first to have the transplant in the United States. But the assurance was that it had been done in Europe before. It just hadn't been done in the States. And there was a gentleman who saw my story, and he called my doctor, and he said, I want to speak to that lady doctor. Her name is Dr. Versacala. He said, I want to speak to that lady doctor. No, I want to speak to that lady doctor. And she did talk to him, and by the way, he did get into the program. He did have the gene that was needed to be eligible for the program. I don't know how he's doing. They don't communicate that to me, but I do know he got into the program. And I just say that to say that it's just important that we communicate what is going on in the area of research. And I'll talk to you about there are venues by which we can also give. And one thing is that African Americans and other minorities are not real big participants in the pool of therapies that are available because we don't get involved in that. There is a program that they do called Be the Match. And Be the Match is one thing that we're not participating in a large number. So one of the things that I've tried to do and will continue to try to do is to encourage people of color to get involved in that because it's not difficult. It's just your genes and your cells can save other lives. And so it's important that we put ourselves out there and be available for those kinds of things. I'm going to close by just saying that it has been an honor to be the first person to have received this transplant in the United States. But at the same time, I know with that comes some responsibility, and I hope that I will have an opportunity to share with others my experience and to be of comfort when people are talking about that. But I do want to give credit to my doctors in the medical profession who helped me to get through this process. I appreciate what the legislators have done in funding the AACR, and I encourage and hope that they will continue to fund it. Those dollars, come from that source of our government that, just can't be matched. And so we really appreciate that. My son, who gave of his unselfishly gave of his cells with love to have me cured. And then to my support systems, my church, my family, my wife and kids who always stood by me during this particular time. And I also liken a lot of to what I went through is my faith in our Lord and Savior Jesus Christ, who helped me to get through this. And I know that the medical professionals are the ones who do it, but I think that my Lord and Savior is the one who gives them those skills. And I believe in that. So I thank you for hearing my testimony today. Didn't mean to keep you that long, but thanks for having me.
Thank you, Chuck and Johnny and your son. And like you said, it's really a group effort. It's not just the patient who's affected, and everyone helps to get you better. And with that said, we have another family here. We have Tori and Dana and Chris who are going to tell their story.
So at 5 years old, our youngest daughter, Tori, had begun kindergarten. She was really excited to go to school, to play sports, and to do all of the things that her 3 older sisters had been doing. But the excitement of that first month quickly vanished when she began experiencing unexplained fevers, repeated ear infections that wouldn't go away despite rounds of antibiotics. We were puzzled, but never in our wildest dreams did we ever think we'd hear the words, "Your child has cancer." Tori was diagnosed with acute lymphoblastic leukemia in October 2008. It was impossible to explain what this was to a 5-year-old. It was excruciating to share the news with her 3 older sisters who didn't know or understand what this meant for her or for our family. We held on to the positive news that this was the most common childhood cancer and the one that had the best survival rate. And so we began the 2.5-year treatment protocol immediately and held on to hope. My husband can attest to this. He was really my rock through all of it because it was excruciating to watch your child be given the toxic chemotherapy and hold her down as the needles came. We were with her day and night as she experienced some awful side effects and spent weeks at a time in the hospital. And during that time, we also tried to be there for our 3 other children, and we felt tremendous guilt for missing their sports, their school, and just everyday life. It was a life that I would never wish on another parent. But we got through the difficult phase of treatment, and we were relieved that the end of treatment was in sight, but it wasn't. After almost 2 years of treatment, Tori relapsed. This was probably more difficult to swallow, as I said in the video, than the initial diagnosis because we knew what we were in store for this time, and we were worried, why didn't it work? We also knew that Tori was going to miss more years of her childhood, but we didn't have much of a choice. A new 2-year treatment protocol was started this time, including cranial radiation along with chemotherapy. And once again, it was agonizing time for all of us. The days, I mean, it was almost 5 years that we were involved in this. They're blurred now, but our lives were turned upside down, and our family was often torn apart. But again, we made it to the end of treatment, and we rejoiced that we were finally finished and Tori was cancer-free. But she wasn't. Tori relapsed a second time, and we were devastated once again and quickly losing hope. Our only option at that time was a bone marrow transplant, but the thought of that grueling procedure scared us as Tori had been through over 4 years of harsh treatment, and we weren't sure what her little body could take, but we didn't have a choice. So while we were preparing for the bone marrow transplant, we were at the Children's Hospital of Philadelphia. We learned about a new trial that was underway there, CAR-19, as Dr. Jaffe said, CAR-T therapy. We learned it was for children with Tori's type of leukemia who were not able to get into remission. And as we read more and we learned more about this trial, we knew that we were determined to have her be a part of it. It was a groundbreaking but still experimental treatment. It is now what is known as CAR-T cell therapy. As Dr. Jaffe explained in her remarks, they remove the patient's T cell from the body and engineer them in a lab to become cancer destroyers. When the engineered cells are infused back into the body, they then search out and destroy leukemia cells. No more chemotherapy, no more radiation, no more horrible side effects. We knew that her leukemia was resistant, and we just knew we had a better chance with this cutting-edge treatment, and we were determined to give our daughter the best chance at survival as well as quality of life. So we met with doctors, and we discussed at length the pros and cons of a bone marrow transplant versus this new trial, and we decided the trial was for us. We eagerly signed up, and we were re-energized by the hope of this new approach because what we had been doing hadn't been working. In April 2013, after 5 years of fighting cancer, Tori became the 10th child in the world to receive her engineered T cells, and the results were astounding. A month later, she was back in school. She has never missed a day of school because of cancer since. Today, she is 5 years cancer-free. She's turning 15 this month. She's just started her first year of high school. She's missing day 3 today. She plays on the tennis team. She hangs out with her friends. She's doing all the things we could only hope for her. And we sit here today because of the advancements of immunotherapy. These advancements would never have been possible without decades of funding for basic research into the immune system, translational research, and clinical trials, most of which was funded by the NIH. I truly don't believe that we would be sharing a happy story if this treatment was not available to her. And we thank everyone who believed in and supported the research necessary to save her life and so many others. Thank you.
Thank you, the whole family, and I think one of the words we never use, or we don't use enough, is brave, and thank you for being brave and going through what you did, and we're so happy you're doing well. So our final speaker is Lori Trotman, and she's going to tell us how cancer survivorship has changed how she thinks about life, I believe.
Thank you. Hi, everyone. Can you hear me? I was diagnosed with breast cancer in 2008 on my very first mammogram. I had turned 40, and my doctor suggested getting a baseline mammogram, and it was there front and center. It had been there for a couple of years, apparently. I was, of course, in shock. I couldn't understand how I had breast cancer at such a, quote, young age, and discussed treatment options with my surgeon. We decided to do a lumpectomy, chemotherapy, and radiation, and I was also required to take tamoxifen for 5 years after treatment. After going through the treatment, the surgery, the first question I asked the doctor was there any node involvement? And there was. So they did a full lymph node dissection, and I've got lymphedema that I've been battling ever since. And then my second question to her was fertility. I was a single professional working in New York, very career focused, did really well, loved what I did, but I wasn't married, and I wasn't in a relationship, and I wanted to know how I could have a baby, and I was 40. And I just kind of thought it would always happen, but it didn't. So she said, go see this fertility preservation specialist at NYU Medical Center, Dr. Nicole Noyes. She helped me have my own twins. And when I went there, it was the best referral I ever had, and they literally moved heaven and earth to get me in there. I only had 6 weeks between surgery and starting chemotherapy. So when I called, they basically said, she's got a 3-month waiting list, but because I was a cancer patient, they literally got me in. It was a Thursday, and I got in that Monday morning. Amazing, the most amazing referral, and I'm personally so grateful for her. We were lucky. We got to do a full cycle to preserve my eggs before I started chemotherapy. I had a great response. I had 20 eggs that they were able to extract and use and freeze for a later date, hopefully. But they were eggs. They weren't fertilized embryos. And at the time, that was considered experimental because most people only fertilized embryos, not just eggs. So when I had to, in order to get ready for the retrieval, I had to give myself injections 3 times a day. And it was a little scary the first time I did it, and I remember I would happily display the soda bottle with the used syringes on the dining room table, and one night my aunt comes by and says, oh, how do you do that? How do you give yourself shots? And I'm like, because I really hope to have a baby one day. So I'm gonna try, And when they went to retrieve my eggs, the anesthesiologist actually said to me, don't worry, we're gonna save your eggs for a better time. And at that time, I realized there was hope for a better time, even though I had no idea what lay ahead. I was terrified of chemo, I was terrified of treatment. So I ended up preserving my eggs. I went through treatment, and one night I was styling my wig, 3:30 in the morning, because I was a little hyper on some of those drugs. My sister came in, and she says, what are you doing? And I'm like, oh, hi, I'm styling my wig, and then we started talking, and I said, I really want to have a baby one day, but if I have a baby as a single mom, and the cancer comes back, and I die, and the baby's alone, what do I do? She goes, but what happens if you live another 30 years and the cancer doesn't come back? You'll wish you had tried. And in that moment, I realized, I'm gonna try. So I grew stronger. When treatment was over, I had severe radiation burns, and I really wish I could have benefited from the 2-week treatment instead of 6 at the time. And I had these radiation burns, and they were pretty severe, and it was a hard recovery process, but I got through it. And as I got stronger, I started thinking about the baby. I started thinking about what could I do. So I could tell you a whole bunch of stuff, but to cut the story short, because we don't have a lot of time, I ended up trying IVF twice. With donor sperm. I forgot to tell you about the donor sperm. This is one critical part. You can buy anything online. You really can buy anything online these days. It's pretty amazing. I found the best donor sperm for me. Smart, sexy, hot, like all of the above. He's really cute. And you get lifetime photos, so this is the best part. So you can kind of select, and we actually have a whole donor family now. My son has tons of siblings, but anyway. I forgot that part, and it was actually the easiest part and one of the cheapest parts of the process, by the way. So I tried IVF twice. I tried for the first time in March of 2011, and it failed. I was devastated and depressed, and I was really scared because I only had one try left. I only had enough eggs for one try, and the second time around, my oncologist and my fertility specialist spoke, and they said, my fertility specialist wanted to give me a little bit of an extra hormone that would make my uterus warm and fuzzy the second time around. And I was banking on, a good pregnancy because I come from a fertile myrtle gene pool. My mom had 7 kids. My sister never had any trouble getting pregnant. So I was hoping that would parlay to me, and it did, thank God. So I did get pregnant with twins the second time around. Unfortunately, I lost one baby in the first trimester, but my son Tristan was born April 5th, 2012. He's now 6 years old. And my very own miracle, personally. I know everyone says that. But without the advances of science made possible by NIH funding and the fact that better treatments are allowing some patients to live longer with cancer, a story like mine would not have been possible. My gratitude knows no bounds. I hope that together we can continue to support cancer research and eradicate this deadly disease and help people dealing with a diagnosis have a chance to live a better life. We all make choices. I chose hope despite my fears. And really, there's nothing better than a dream coming true. This I know.
Thank you so much.
So we just have a few minutes left, and I wanted to ask a couple of quick questions. First for Dr. Lee. So you talked a little bit about the liquid tumor biopsies. How do you see that moving to practice where patients in clinical care will be taking benefit from that?
Yeah, so there are several clinical trials right now looking at, for example, we treat the patient with one-size-fits-all. Everybody gets 6 cycles of chemotherapy or 6 or 7 weeks of radiation, depending on what type of cancers you get, or 2 weeks of radiation for breast cancer. The question is, first, can we actually figure out whether they need less or more, the radiation or chemotherapy? And then right now it takes us months of an imaging study to figure out whether the tumor is actually responding to the treatment we're giving, and they get all the side effects that they're receiving. But can we be smarter than that? Can we take a look at it with 1 week with a liquid biopsy into the treatment And if they don't benefit from the treatment, why do we continue for another 6 months? Why can't we move it sooner? So those are the way that we're looking to optimize our current therapy. And then obviously, if we can use them for detection earlier, that's even better, because when we detect the tumor much earlier, we need less treatment, we have less side effects, and patients go back and higher chance of cure.
Great. So do you see this moving into the clinics in the next few years? Will we be talking about it next year in the report, or?
You know, clinical trials does take us some time, and some of that has to do, I think that the detection studies will come out sooner, but I think the one that related to treatment and modifying treatment, obviously if you have a situation where you're treating for cure, you're a little bit more reluctant to change the treatment because you can potentially cure versus not cure. And so there's a randomized study going on, and I think those will take a couple of years.
Great, thank you. So I just wanted to ask each of the survivors a question. So with everything you've gone through, and you've certainly gone through an awful lot, what is the one thing you wish that you had known at the time of your diagnosis? Start with Lori.
I wish medical advances were more advanced, even, because at the time they did this procedure for the sentinel node biopsy where they do some dye during the surgery to figure out if the cancer spread to a node, and in my case it had the only spread to the sentinel node, and they ended up doing a full dissection, and everything they took out was healthy after that. So I wish, some of the medical advances were available so that I, would have not had to have unnecessary surgery.
Thank you. Chuck, what would you say?
I wish that I had known that the treatment that I was going to receive would be one that would give me a cure, but at the same time not have to go through it. I mean, it's
that's true.
You know that it's a cure. You know that the trials are there, but I was hoping that I wouldn't have to go through a trial like that to cure me. But I will say this, and that is that I'm so glad that it was there, and it did cure me of the disease. I'm almost 3 years from my transplant, and so it just wasn't much that the doctors didn't really inform me about, and I guess I was pretty well informed.
That's wonderful, thank you.
Yeah.
And Tori or Dana?
There's always hope.
Yeah,
there's light at the end of the tunnel.
Yeah, I think when you say what we could have known, if we had known we'd be sitting here in 2018, it would have helped a lot. But I think you don't really understand. I mean, now we feel like we're experts in the world of cancer, but you don't know what it all means, and you learn a lot. And it's a very long journey with the unknown. So if I could have looked ahead and seen this, I would have made it a lot better.
Do you feel that knowing that there was good clinical research available gave you hope?
Oh, absolutely for us. I mean, with her 2 relapses, we, she wasn't following that path that they had told us, it's the most highly curable cancer, and she broke every rule on the journey. So I don't think, and it was timing too for us, because had we not gone through those things and taken so long to get there, this might not have even been available to her. So, it's kind of like the best laid plans, and we were very fortunate.
It's wonderful.
To be involved.
Well, I want to thank all of the participants, including, let's give the survivors an extra round of applause. And I think you all certainly gave us a lot of hope and reasons to continue this fight against cancer. I want to thank all of you for joining us, and I especially want to thank the American Association for Cancer Research, leadership, staff for putting this event together, but importantly for putting the report together every year. Thank you very much.